Okay, back again. We've got a slight change in the program now. Unfortunately, our established the program speaker couldn't speak, but thankfully Lisa from All the Biosciences shifted her slot. And so what we're going to hear today is quantifying quality, key bit in alternative protein. Quality is killer in terms of products. So let's look at advancing alt proteins through amino acid analytics. Lisa, it's all yours. Looking forward to this.
Thanks very much. So yeah, it's a different angle from most talks at these sorts of meetings, because it's really important to really sort of address the types of work that you might need to do during your product development or for regulatory requirements. So yeah, I'm the managing director of Alta Bioscience, and yeah, we are a analysis laboratory and a custom reagent synthesis laboratory as well. So we've actually been established for over 50 years. For 20 of those years, we've had our ISO accreditation, so 17025 with UCAS. So this is the required laboratory accreditation for our amino acid analysis and our N-terminal protein sequencing. So we have very fast turnaround times for analysis, and you also have sort of experts, technical experts that you can talk with directly to give you advice and guidance around the kind of testing that you might need, and some of the sort of the intricacies around the sort of sample processing and sample volumes that we might need as well. So really, I want to talk to you now about sort of all the different areas really around amino acid analysis. So this sort of breaks down to the services that we offer. So mentioned the protein sequencing, amino acid analysis. We also do things like mass spectrometry, where you might want to sort of look at sort of the molecular weight of certain proteins, and sort of identify certain proteins as well if you're doing sort of fermentation, so protein production. One of the newer sort of additions to our service offering is in vitro protein digestibility studies. And I've got a few slides at the end of the presentation around that as well. And then other areas of the custom services. So where we have sort of peptide synthesis, maybe more for using peptide scaffolds in cell culture for sort of the sort of cultivated meat industry. And then some other areas include sort of oligosynthesis and antibody generation, which is probably more for some of our biotech customers. So to go really into the sort of types of amino acids. So many of you are probably familiar with this. So amino acids are the building blocks of our proteins, play a fundamental role in the growth, repair, and bodily functions. We have the 20 standard proteinogenic amino acids. So this compiles of the 9 essential amino acids. So these have to be obtained from the diet. So it's really important that they're in sort of the right levels in the food that we're eating. And then you have the 11 non-essential amino acids. So these are those that are produced by the body by itself. And then we have a lot more non-proteinogenic. So we have over 100 different types of other amino acids. So these aren't directly incorporated into proteins, but they do have a role in the metabolism, in the signaling and the physiological functions in the body. So I've just given some examples here of the amino acids that might be familiar to you all. Things like taurine is a key amino acid in cat food, because that's something that cats can't produce, but it's really important for them. Hydroxyproline is one of the amino acids that looked at when you're looking at collagen analysis. So really there's a sort of a whole breadth of different sort of amino acids that you need to sort of be looking at potentially. So I guess the question is, why profile amino acids? Why not just look at sort of total protein? So looking at from a nutritional perspective, so it really gives you amino acids of quantitation will give you an accurate protein concentration. That might be important for labeling and quality control, but also giving you an indication of what that protein quality is. So your essential amino acids versus your non-essentials. It might be for health and nutrition claims. So in the sports food sector, it might be that you're wanting to sort of say something particularly high in a particular amino acid for muscle repair. Or for other health claims, you might want to have certain amino acids in there for that. So another area would be product development and quality control. where you are wanting to look at developing formulations for things that might sort of influence flavor, the texture, the mouthfeel. You might also want to look at sort of where you're optimizing the manufacturing process. So this is areas include such as sort of cell culture of sort of the cultivated meat industry, where you're wanting to look at how the uptake of amino acids is affecting the growth of those cells, and then sort of going into sort of precision fermentation. Again, what is it that sort of what's the impact on all the amino acids in that mixture and the sort of final protein that you're producing as well. It could be that you're wanting to understand the product safety and making sure that that product is consistent from time to time, so you're getting that accurate analysis. Examples might include sort of the analysis of asparagine, which is sort of part of the risk of looking at sort of acrylamide production in food from sort of baking of say of certain goods like potatoes and so on. And ultimately, it's a regulatory compliance. For many working in the novel food sector, there will be a need to have the amino acid analysis done as part of your regulatory dossier. So some of the applications, and I've got some sort of slides later on about some of the examples, but it's really varied. You've got anything from sort of the waste byproducts, turning those into new protein sources, mentioned about cell growth optimization. Maybe it's changing away from animal-related materials, so bovine collagen for alternatives, maybe marine-based collagen. You've got the fetal bovine serum, again, finding alternatives to that. Looking at cultivated versus conventional meat production, and really sort of understanding the accurate and reliable data. So looking at product consistency, whether it contains certain amino acids. So there's really many different applications that you might want to get this really in-depth profile. One really interesting area that we're seeing a lot of at the moment is the interest around the impact of amino acids and flavor on your foods being produced. So this sort of table gives you, or these images give you an idea of some of the impacts of certain amino acids on the flavor profiles. So you can see, for example, alanine might have a sweet influence, asparagine has a bitter influence. So if you're trying to sort of look for a blend within a food to give certain sort of features, certain taste profiles, then you can look to sort of manipulate that accordingly based on these sort of impact of each of these types of amino acids. So yeah, glutamate and aspartate have a strong enhancer and umami flavor. You've got the branched chain amino acids such as valine, leucine and isoleucine. They will give you those kind of malty, fruity flavors. And then you've got glycine, can be used to mask any sort of off flavors in food and beverages. So it's a really sort of interesting angle to sort of look at how we can provide you that sort of really quantitative and sort of non-subjective approach to sort of the tasting profiles. So when you look at sort of how traditional protein quantitation is done, when you're not looking at amino acid analysis, the Kildall and Dumas methods are the most commonly used. Now, the advantage of those is they are very simple to do, very low cost, and they can be very quick to sort of turn around those results. The downside is that you are measuring total nitrogen and actually not the true protein in that sample. So you'll have variability in different conversion factors, and it will overestimate sort of protein content in many occasions. So you're not really getting any information on the quality of the protein, or in fact the amino acid composition. So what you do get from amino acid analysis would be really sort of a more sort of superior method where you're getting that accurate quantitation, you're getting the composition, so you will get a complete breakdown of the concentration in that protein to give you that sort of total protein at the end, and you'll be able to see that profile. Highly sensitive, so you'll be able to get very low levels of detection, So it's important, if you're actually looking at low protein foods rather than high protein foods, you'll be able to sort of really understand the level of each of those amino acids in there. And it's also very highly reproducible as well. So you don't have to worry about sort of those conversion factors that you might have within sort of looking at total protein from the other methods. There are two core ways that you could look at amino acid profiling. You've got the total amino acid analysis. That would be where you're quantifying the total protein from the bound and from the unbound amino acids. So it would be peptides and proteins in that sample. And what happens, you're then hydrolyzing that entire sample to break down all of the protein into its constituent amino acids. And then you have your free amino acid analysis, which is basically anything that's unbound within that matrix. So you would have your proteins and peptides precipitated out, and you're just looking at what would be the free amino acids in that material. So it's really important when you're looking at the methods to analyze amino acids. It's not just about sort of the analytical technique, it's also about the sample preparation. And actually this is one of the hardest parts, to really make sure you have a really rigorous and standardized protocol to ensure that those results are accurate and reproducible every time, and that you're able to quantify all of the amino acids of interest at the right level of recovery. So during the protein hydrolysis, for the total amino acid analysis, we have to do the protein hydrolysis. So we start off with the grinding, homogenization of the sample. So obviously you can imagine all the different types of materials that you might have to look at. So it could involve sort of freeze drying the sample, grinding it up, or just sort of mixing it up literally in a blender. So we need to make sure that you've got a really thorough and uniform disruption of that sample. But what you don't want to do is heat it, because by heating it at this stage, you could actually destroy some of the amino acids and obviously that is something you want to completely avoid. So you would be able to get sort of the complete protein extracted at this point. Then there are certain amino acids that are quite sort of sensitive to the process. So things like tyrosine and methionine, we need to protect them to give maximum recovery. So the method that we use in particular is degassing of the sample under nitrogen. And we also do a sort of flame sealing of the tube that we have the sample in. And this is really important to protect those two, and it gives us very good recovery. I think other methods might have to compensate for an element of loss of those materials, but we want to maximize those as much as possible. So it's really important that we use this sort of, it's quite a manual process, but it also is highly accurate. And then you have considerations whether you're looking at an acid or an alkaline hydrolysis process. So there are amino acids like cysteine and tryptophan that would be destroyed in our standard hydrolysis method. So we have to do those separately, again, to ensure that you get the maximum results out of those. So this just quickly sort of summarizes where we have our standard hydrolysis method. So you'd get 18 of the 20 amino acids during that sort of preparation. With cysteine analysis, we have to essentially oxidize the sample with a particular acid to protect it and to convert it to cysteic acid. And with tryptophan, we have to use an alkaline hydrolysis method, again, to ensure maximum recovery. For the free amino acid analysis, it's slightly simpler. We don't have to obviously go through that whole hydrolysis process, but what we still need to ensure that we're getting that sort of standardized protocol. And this time we're looking at 40 amino acids in this run. So it's a very long, very sort of well separated method. But first of all, we need to do the protein precipitation. So we're removing the peptides and the proteins from that sample. And then you need to do the sort of sample dilution. So it might be that you're wanting to make sure we're getting full accurate quantitation across all of the amino acids, some that might be in higher concentrations than others. So we're wanting to make sure that you have those results are within the linear range of the instrument. Obviously, you can look at many different sample types. So we might have peptides, recombinant proteins, complex matrices, physiological fluids. So if you're working in sort of the sort of the human sort of trials of products, you might want to look at serum samples to see, sort of the influence of the protein diets that people are eating, and to sort of see what impact it has on them from there, looking in their plasma and serum. And it could be looking at looking at the spent media and biological samples as well. So really a variety of different things. And sample volume, I know, is very precious to many. So we can actually use a very low volume of sample. So it might be a gram of dry samples or 2 ml of solution. And if you've got a pure protein, then we only need 5 milligrams of that to be able to do the analysis. So, and even with that, we can then get very low detection limits. So as low as 10 picomoles. So yeah, really sort of don't need a huge amount of sample, which I know is really important in your early development stage. So the method that we use is HPLC with post-column derivatization. So we do the hydrolysis at the start, or the total, that's for the total amino acid analysis, obviously for the free with the precipitation. We then do the separation of the amino acids on an ion exchange chromatography column. And then we derivatize with ninhydrin before we then go through the detection stage. And the importance of this is that actually by doing post-column derivatization, you're removing a lot of the interference that may be remaining from other compounds within the sample. So that helps us get very clean baselines and very good quantitation and very good low levels of detection as well. Here's an example of what you might see. So I mentioned sort of the 40 amino acid run that we have. So this is we class as our sort of high resolution method. So you can see really nice separation of many of the peaks. We've got sort of all 20 proteinogenic amino acids in this. And we also have 20 of the non-standard amino acids that I mentioned previously, so the likes of taurine, GABA. And we can also look at other, essentially anything that reacts with ninhydrin. So we can look at those sort of ninhydrin positive substances like ethanolone, I'll forget that one because I can't say it. So here's some examples of some case studies that we've worked on for customers, and just gives you a flavour for really what is possible and it might be sort of applicable to your industry. So collagen analysis with looking at sort of hydroxyproline. So Gelagen are looking at sort of marine collagen as an alternative. You have those vegan cowboys that are looking at casein produced through fermentation process, and they were looking at being able to create a set of their own standards for their QC process, and they wanted to basically make sure that their standards were accurate. So that sort of required a lot of testing to be able to sort of get a set of standards that could be usable. And then you've got looking at in the cell cultured meat, we have Meatly, where they were doing the comparison of the product they produce with standard traditional products, and also looking at during product development as well, looking at those profiles of amino acids during the early development phase. And then James Robinson Specialty Ingredients were looking at the use of a novel ingredient and how that hydrolyzes and releases and protects certain amino acids that be important in sort of the cosmetics industry. So a little bit about protein digestibility. So why would you want to look at this? So it might be important for looking at how efficiently these proteins are broken down and absorbed. Understanding nutritional quality of a protein, and really sort of give guides on nutrition recommendations, as well as identifying effective protein production on digestibility. There are a few methods to be able to do this, but the areas we're focused on in particular would be the static and the dynamic in vitro methods. So I think as we're running probably a little bit short on time now, I'll quickly go on to sort of the last couple of slides, really just to sort of, we can obviously discuss this in more detail if you have any additional questions later on, but essentially you're looking at the oral phase, the gastric phase, and the intestinal phase when you're looking at the in vitro digestion model. and being able to combine it with amino acid analysis gives an extra layer of data rather than just looking at total protein. So the studies will give you digestion profiles, understanding the extent of digestion of that protein, quantitative data, and the rate of absorption as well. So I think with that, hopefully that's,
Great.
Thank you very much for your attention.
Okay, so we've got a couple of questions coming. One from Kelsey Kanyuk at This company, some great products they produce. She said, do you see the main reason for companies looking at testing of the amino acids? Are they consumer, regulatory, product confidence, something else, or all of the above?
All of the above, definitely. So there's a regulatory drive, absolutely. If it is a novel food, that we see a lot of companies wanting us to provide a lot of detailed information on the methods used and a lot of samples run to get that data. I think consumer confidence is a really interesting one, because we've spoken to companies where they're wanting more than just the sort of marketing claims. They're actually wanting to see data. I think that's really important. So it's definitely all of the above.
Okay, cool. I suppose the thing, because you're providing services, I mean, how quickly can your lab generate this actionable data on fast product? I mean, everybody wants their data yesterday.
Absolutely. So we claim a standard turnaround time of 7 to 10 working days. Then we have sort of an express option of 5 days. So to give you some context, it actually, because of the process I've talked through, from the sample preparation all the way through to those really nice sort of resolution peaks during the analysis, that process itself can take 3 days total from start to finish for just one sample. So that's the quickest we can ever do it. But obviously we work with customers to sort of see what their timelines are.
Okay, and we're seeing evolution of the digestibility testing. I mean, the in vitro models are getting better and better. I know the MyGut model and Kier2 that in Leeds. How do you see that evolving for next generation proteins? Do you think it'll become an essential part of the kind of triage testing thing?
Yeah, I think it will. I think speaking to a lot of the regulators, or the consultants, I think there's going to be a lot more focus on looking at those sorts of studies. I think it's also making sure that the studies are appropriate for that particular industry. So if it's say aquaculture, that you're producing your feedstuffs for, that it's the right model for that particular area, and making sure again that the protein that's being used as a comparator is the right kind of protein. So it's having the right model for the right food and the right industry, I think. So I think there's going to be a lot more regulations around that as well.
Okay. Interesting question, this one. We'll make this the last one due to time. How do you compare precision fermented proteins from a nutritional point of view as an ingredient or formulated end product? I'm entirely sure what I'm getting to there. I mean, to my mind, presumably digestibility is digestibility, and the company makes their decision on that. You just provide the data, yeah?
Yes, exactly. From our perspective, yes. I mean, when you're looking at, from the digestion perspective, we're looking at just that sort of amino acid availability, essentially, of that protein, what's available. And I didn't touch on the absorption side of things, but obviously there's people working in the absorption space as well. So it's really about understanding what amino acids are available. So yeah, how you compare it to kind of the production process, I think that's yeah.
Okay, so there's some other questions. What I'd suggest, on interest of time, just contact Lisa directly. The email's there. There's the backstage part. But thanks again for that. It's hugely informative, and a bit that actually people probably initially don't think they need, and then they start to scale up and they realise you absolutely need this to go forward. So thanks for that, Lisa.
Thank you very much.